Hello everyone, in this video we want to continue talking about the making of protein. At the beginning we said the DNA will copy the information in the process which is called the transcription process into messenger RNA. Then the messenger RNA will be released through the nuclear membrane from the nucleus to the cytoplasm. So imagine with me now, this is the mRNA that is copied, and it has the information that are copied from the DNA. What is the next step? The next step is what? Is a translation, to translate the information in the mRNA to make the protein. To make the protein, what are the things that we need? We have the ribosomes. You remember the ribosomes? The ribosomes, their function in protein synthesis. What is the ribosome? Is made up of a protein, and it's made up of protein. Just hold on. So the ribosome is made up of protein and ribosomal RNA. Now the function of ribosome, the second type of RNA, which is ribosomal RNA, it comes. So here for the ribosome, it has two units. I want you to look at this one. This one here, it shows what? It shows the ribosome, how it start to attach with the mRNA. This is the mRNA, just to show you. This one here is the mRNA, this thread. mRNA. Sorry for this. So this is what the mRNA, which is here. Okay. What is the mRNA? This one is the mRNA. This is thread. Okay. Now the ribosome, which is made up of a protein and RNA, ribosomal RNA. This is the ribosome. The ribosome will come and will attach with the mRNA. The ribosome, it's made up of two units, the upper unit and the lower unit. I don't want to talk about them, but I want you to know now how this works to make the protein. You can imagine this ribosome, it's like, you know, when you put a bead inside a thread and you start to moving this beads, the same thing. Okay, now this ribosome will come here, and the other molecule will come, which is a transfer RNA, tRNA. What is that tRNA? I'm going to show you first the structure of the tRNA to understand how it will work. Imagine with me, this is the tRNA. It will come. It carries with it, it has at the top of it, it carries what? Here, an amino acid. You know what is the amino acid? Here it has amino acid. And this one here, it has what? Let's write it here. This one is called amino acid. And here it has anticode here, like 3 codes, 1, 2, 3. These anticode, and for this anticode here, what are they carrying? Just hold on, just to show you. And it has, like here, it has at the bottom, let's go back, anticode, let's say it's G. Here I'm discussing what? The structure of tRNA. So what? Why? What is the tRNA? We were talking about the mRNA and the ribosome. What is the function of tRNA? This tRNA, it carries what? Amino acid, and this is the anticode for this amino acid. Okay, you know the amino acid, which is the building block of a protein. And here, if we want to go back, this is the ribosome. This is here the tRNA. It's carrying with it amino acid, and this is the anticode of the amino acid. It will come, this transfer, and it will attach with the nitrogen bases here, with the 3 nitrogen bases on the mRNA. Each 3 bases of nitrogen bases in the mRNA, it's what is a code for what? For amino acid. Yani for example, I'm going to show you this figure to understand this one. Look at this figure. This is what here, this is the blue thing here, which is mRNA, messenger RNA. Okay? And this one here, which is the ribosome. It will attach to mRNA. Okay? And this one here, which is the first tRNA, will come. This is, I'll put on here number 1. This is tRNA. It carries amino acid LYS. Okay? For LYS, the anticode of it, U, 3 U's. So, what is the 3 codes in the mRNA? U attached with A, U attached with A, U attached with A. So, it will first make the first amino acid. Another amino transfer RNA will come, tRNA will come. This one, this code, it has CUA. CUA it will be linked with GAU, and this one for ASP amino acid. This is the second amino acid, and so on. Another a tRNA will come, and these amino acid will be linked together. So to make what? To make the specific protein. So this is the way how the tRNA with mRNA with ribosome it has ribosomal RNA are working together to make the translation process. This is the process of making protein and attach these amino acid together. Here actually the tRNA transfer its amino acid to a growing chain, then the tRNA is released. It will be removed. It stopped. It's finished. Okay, it works. It's finished. Okay, so it released. The ribosome moved down the mRNA and the process repeated. You understand? When the ribosome reach the end of the gene, the chain of amino acid is released. This is the process of translation process. As I'm going to repeat it from the beginning. From the beginning, during transcription, DNA is used as a template to make a complementary strand of mRNA. Here in eukaryotic, the mRNA is exit in the nucleus. Here the transcription process at the beginning that we studied in the previous video, it's in the nucleus. Then this mRNA, it will be released to the cytoplasm. The mRNA, it will attach with ribosome. Ribosome here, what happened? The tRNA will come and it will attach with what? It has, this is the anticode, this part. And this is the amino acid. It's for this amino acid, and it will attach with what? With the messenger RNA. Then another tRNA will come, another tRNA come, and the ribosome is moving through the mRNA to make the full chain of the required protein. So a ribosome attached to the mRNA strand, the beginning of the gene. The tRNA molecule enters the ribosome. It will enter the ribosome. Okay, 3 bases on the tRNA match up to 3 complementary bases on the mRNA strand. The bases on the mRNA strand determine the tRNA, and amino acid move into the ribosome. Okay, so then the tRNA transfer its amino acid to the growing chain. Then the tRNA is released. The ribosome moves down the mRNA, and the process repeats. This one is, if you understand this figure, it's very helpful to understand this translation process. Once the ribosome reaches the end of the gene, the chain of amino acid is released. This process, ribosome and tRNA, and the ribosome, it has the ribosomal RNA. This process of making the amino acid, or making a growing chain of amino acid, or to making the protein, here it's called what? Translation process. So here we finish from transcription, then translation. So let's go back to the definition of translation. The information of mRNA is then used to build up the protein. This one is called translation. So in translation, the mRNA passes through a protein assembly line within a ribosome. And just to remind you, a ribosome is a cell organelle made of ribosomal RNA and protein. It has 2 parts, ribosomal RNA and protein. As mRNA passes through transfer RNA, tRNA, which is the third form of the RNA, okay, delivers amino acid to the ribosome. The order of bases code for the amino acid is attached. The amino acid are joined together to form what? A protein, a long chain of amino acid. It will give the protein. So this is the process of making what? Of making protein. I know it's not easy at the first time when you hear about it, but if you repeat the information step by step, you will find it easy. If you still have a question about the translation or transcription process, you can send me your questions. Thank you for listening.